Hello dear friends, welcome back to my YouTube channel. Today here I am going to discuss about vaccination. Let's see the definition. It is the process of stimulating protective adaptive immune response against microbes by exposure to non-pathogenic forms of components of microbes. So development of vaccines against infection has been one of the great success of immunology. Only human diseases to be intentionally eradicated from the earth is smallpox with the help of vaccination, and this was achieved by worldwide program of vaccination. Nowadays polio is also likely to be the second such disease that have largely controlled by the vaccination. Apart from this, there are several types of vaccine that are used and being developed. So vaccination is an effective strategy for restraining the infection. Remarkably, potent vaccines are those that are successful in provoking high affinity antibodies and memory cells. And most of the vaccines develop function by inducing humoral immune response in the host. There are different types of vaccines like attenuated and inactivated bacterial or viral vaccines, purified antigen subunit vaccines, synthetic antigen vaccines, live viral vaccines involving recombinant viruses, DNA vaccines, adjuvant and passive immunization. These are the types of vaccines where we'll see in detail in upcoming slides. So attenuated or inactivated bacterial vaccines, where vaccines are containing intact non-pathogenic microbes, are made after attenuating the virus or killing the microbe. So this is a virus which is attenuated, means its infection is minimized and it has not infective virus. So they are effective because they are not infective, but when they enter inside the human body they are evoke effective innate adaptive immune response. So bacterial vaccines offer the vaccines which are made by bacteria. They offer protection for small duration, while the viral vaccines elicit good response and long lasting immunity. However, the viral vaccines always face important risk of reversion to virulent, and hence safety is the main concern for such type of attenuated vaccines. To minimize such risk, inactivated vaccines are used, such as influenza vaccine. So BCG, typhoid, oral plague, oral polio, yellow fever, measles, mumps, rubella, influenza, and typhus are the examples of attenuated or inactivated bacterial vaccines. Second type of vaccine is the killed vaccine or inactivated vaccines, where the virus is killed and the killed virus is, that is inactivated virus, is directly inserted. So vaccines are containing killed or inactivated microbes are called as inactive vaccines. Typhoid, cholera, pertussis, plague, rabies, salk, polio, intramucosal influenza and Japanese encephalitis. These are the examples of this type of vaccine. Third is the purified antigen vaccines, where a part of the virus is or component of virus is inserted as a vaccine. Subunit vaccines are those contain a purified antigen and also need to be given along with immunogenic enhancers called adjuvant. They are constructed by isolation of specific protein from a virus or bacteria before administration. Diphtheria and tetanus are the best examples. Or along with this meningococcal polysaccharide vaccine, pneumococcal polysaccharide vaccine, hepatitis B polypeptide vaccine. These are the some example for this type. Synthetic antigen vaccines are, this concept involves identification of epitope for an antigen and designing it in laboratory to be used in future as a vaccine. Next one is the live viral vaccine, where are also called as a recombinant virus vaccine, where a DNA from the virulent virus is purified and then that is inserted into a new vector that is non pathogenic one. Okay, so this DNA, this involves the insertion of a gene that encodes antigen into a non cytopathic virus, which provides immunity following to its instruction to susceptible hosts. They offer protection by eliciting both innate and adaptive immune response. However, in some cases safety is the issue. Hepatitis B vaccine, smallpox, variola vaccine, these are some examples of live viral recombinant vaccines. Next is the DNA vaccine. They prepared by isolating the DNA and injecting the DNA or RNA. Contain plasmid DNA. Okay. They elicit both humoral and cell-mediated immune response without any argument administration, but they their effectiveness needs more experimentation and verification. Adjuvants are the immunogenic substances that do not provide immunity by themselves but enhances the immunogenicity of the vaccine to which they are administered with. And last one is the passive immunization. It is another way of providing protective immunity. This is done when you need urgent immunization. This can be done by transferring a specific antibodies that are useful in cases like snake bite. Passive immunity does not offer long lasting memory, but it is a short lived and does not great memory. Thank you. If you find this video is useful, then like the video, share with your friends, and subscribe to my YouTube channel for watching more upcoming videos. Thank you.